So the last video in lecture 1 is about bacterial phyla you must know. So these are big groups, and what I want you to have is some idea of what they are. So what makes them unique, what makes them special, what makes them really different from the other bacterial phyla, and more than that, what are 2 examples you should know. So I will ask you, tell me 2 genera from a phylum. I'll tell you the phylum. And you'll write down genus number 1 and genus number 2. So here we go. The Firmicutes. This is a really abundant group of gram-positive bacteria that we know very well. So they're well studied and well known, like bacillus. There are lots of different bacillus bacteria, like Bacillus anthracis causes anthrax. Bacillus subtilis, food poisoning, but it's also a really well-known model organism. Clostridium, these are the bacteria that cause, pseudomembranous colitis, Clostridium difficile, but also tetanus, Clostridium tetani, botulism, Clostridium botulinum, and there's one more. Oh, gas gangrene, Clostridium perfringens. It also causes food poisoning. These are 2 really important genera for other reasons that you will learn about in the next lecture. And then these are 2 that we'll see again and again and again because they cause lots of infections in humans, or lots of diseases. Also, this big group has things like lactobacillus and lactococcus that we find in yogurt and cheese and kimchi and sauerkraut. So a lot of the good bacteria, the probiotics that people pay to eat, are from this group. If we find large numbers of Firmicutes in the human gut, that's associated with obesity, and we are not yet sure what the connection is between those 2 things. But there you have it. I tell you that, but don't act on that information. There is nothing we know you can eat. There's no probiotic we know of that will push a person strongly towards or away from obesity. This is an example of a member of the Firmicutes, Staphylococcus aureus. This is a gram stain. They are tiny little spherical cells that grow in clumps. So this is a clump of like 50 of the cells. And this is under the maximum magnification. This is 1,000 times magnified using a oil immersion 100x objective. This is the best you can get, essentially, in a teaching lab, anyway. And this is a gram stain. These are gram positive, so they show up dark purple or dark blue in the gram stain. Next, Actinobacteria. This is the other gram-positive group. And these tend to live in soil. They grow in filaments, so they kind of look like mold. And these are the ones that make soil or dirt smell like soil or dirt. They are also famous for how many different chemicals they can make, how many different molecules they can make. And they make some of our antibiotics. So for example, Streptomyces makes streptomycin antibiotic. And a lot of researchers are working to figure out ways to get them to make other complicated antibiotics that we can't easily synthesize, like vancomycin. One of my good friends in graduate school is an expert in genetics of this genus. There are pathogens in the group. Mycobacterium, this can cause things like tuberculosis. Cyanobacterium causes diphtheria. And then probably my favorite from the group, Bifidobacterium. This is thought of as a very beneficial bacterium that is typically found in human breast milk. So it grows very well on the sugars that are in that, and This is one of the first things that would colonize the gut of a newborn human that is breastfeeding. Okay, the next group, Proteobacteria. This is a gram-negative group. It's the biggest bacterial phylum. It has the most different organisms in it, and they are really different from each other, but they have a fairly recent common ancestor. It's just weird. They're also really well known because many of them are easy to grow in a laboratory. And so early in the history of microbiology, we were able to study a lot of these, and people got the idea that these were the most common bacteria, when they are a big group, but they're not the most common in the environment. A lot of them are well-known pathogens. Escherichia, or E. coli, there are lots of different strains of E. coli, and some of them do cause diseases. Shigella, I mentioned before. Salmonella cause a lot of different diseases. Neisseria causes gonorrhea. Yersinia pestis causes the plague. Vibrio causes cholera. So lots of different examples we can come up with. The Rickettsia cause things like spotted fever. And the Rickettsia are a distant cousin of mitochondria. So the mitochondria inside your cells, those organelles, share a common ancestor with these bacteria. So mitochondria look like little bacteria, and they have DNA. They have their own DNA with ribosomal RNA genes. And so you can compare mitochondria to bacteria and figure out that they are very similar to the Rickettsia. So that's why we think deep back in history at some point a cell ate a member of the Rickettsia, and it just became something like a mitochondria. So that's wild. Yes, and also Proteobacteria a lot of times are found in our guts. We'll talk about that more. This is E. coli, gram stain. Short little rods, sometimes in chains. That would be like two of them. And they would be pink because they're gram negative. Cyanobacterium, these are the really weird gram negative cells that are closely related to chloroplasts. So those organelles within plant cells that are photosynthetic, they are closely related to cyanobacteria. These are photosynthetic, so they get their energy from sunlight and they get their carbon from carbon dioxide, just like plants do. And oxygen on Earth, the original oxygen in the atmosphere came from these cyanobacteria long, long ago. When I was in high school, I was told these are called blue-green algae, and no one says that anymore. I don't know if you heard that or not, because I don't know what they teach in high schools other than mistakes about the scientific method. But Cyanobacterium is the name for these bacteria. So one example is what the food industry calls spirulina. It's the genus Arthrospira. And another is Anabaena. And you'd find these in fresh water, and they form really weird chains. And all of these are going to be green, and all of these are photosynthetic. Here's where we find Cyanobacteria in the universal phylogeny, and somewhere over here is where we would find Rickettsia, or within the Proteobacteria. And notice these little dotted lines. These are showing sort of when did chloroplasts appear in the eukarya. So when did a cyanobacterium become the first chloroplast? And so if we look at this tree, we're kind of going back in time from the animals to the common ancestor of the animals, fungi, and plants, which is here. Farther back, we find the common ancestor of all of these. We go even farther back, we find some cells, some common ancestor of all of these, that had the first cyanobacterium acting as a chloroplast. If we go even farther back, we find the first cell that had a mitochondria. So it must have eaten something like a rickettsia, and that would have happened back there. So cyanobacteria are here in the bacteria. They are not algae. Algae would be plants or other groups within the eukarya. So this is like algae. Algae are huge eukaryotes. They're beautiful. They are photosynthetic, but they're not bacteria. And this is what I talked about. This is what I talked about. Another group, the Chlamydiae. These are gram-negative. They're tiny, and they live inside our cells. They have to live inside a host cell. And so they are, tricky to study and tricky to eat, or to treat. One common genus is Chlamydia. These cause blindness and the sexually transmitted disease, sometimes called just chlamydia. Chlamydophyla, which should be italicized, sorry about that, is another genus, and it has this species within it that infects birds and sometimes causes diseases in humans. Spirochetes is another weird group. They have this really weird cell shape of these long corkscrews. And some of the key members of this group cause really hard to treat diseases in humans, like syphilis and Lyme disease. And they're strange. So that's the end of lecture 1. That's it for lecture 1. I will post a corrected version where I correct the 3 typos I just found. But otherwise, I'm proud of you for making it all the way to the end of this video. And again, I want you to think really hard about how you're going to spend your time in this class. And I recommend going through the slides first and then watching the videos, because you don't have time to watch the videos 3 times. So please bring your questions to class or ask them on the Q&A forum, and I will see you soon.